The current slide is a flowchart of all the steps that we have discussed so far under downstream processing. Now let's have a quick review that will help you in learning the topic more better. So after the fermentation process is complete, the next step is the downstream processing. And the method that you choose first of all will depend if your product is intracellular or extracellular. If your product is intracellular, that is if it is present in the microorganism that you have used for causing the fermentation, you have to break that organism in order to release the desired product. Cell disruption methods fall into four major categories. First is the physical method that includes freeze thawing, which is a technique that involves freezing a cell suspension in a freezer and then thawing the material at room temperature. Multiple cycles of this will cause the cell to break down and release the intracellular material. In osmotic shock, a cell from an isotonic medium is transferred to distilled water, which is hypertonic, and this will result in a rapid influx of water into the cell, causing it to burst. Mechanical methods that includes liquid shear, where the cells are lysed by forcing them to pass through a narrow space, leading to shearing of the cell membranes. In solid shear, the microbes are first frozen at -25 degrees Celsius and then they are passed through a narrow orifice by applying pressure. In agitation with abrasives, the beads, which are made up of highly resistant materials like glass, alumina, ceramics, are added into a disintegrator along with the sample. This is a highly efficient method where 85% of cell disruption can be achieved. In ultrasonications, the cells are subjected to ultrasonic vibrations that ultimately lead to shock waves that will disrupt the cells which are present in the sample. Pestle and mortar can also be used for crushing the samples after freezing them in liquid nitrogen. In chemical methods, detergents disrupt the structure of cell membranes by solubilizing their phospholipids, disrupting lipid-lipid, lipid-protein or protein-protein interactions, which will ultimately lead to release of intracellular content. Solvents solubilize lipid components from cell wall and denature its protein, which leads to release of intracellular components. Alkali treatment includes use of sodium hydroxide, which solubilizes the phospholipid and protein components of the cell membranes. Biological methods includes use of enzymes to remove the cell walls, thus preparing the cells for disruption or for preparing of protoplasts, that is cells without cell walls. Enzymatic treatments are usually carried out in combination with detergent or osmotic shock or mechanical disruption methods. This is because it will ultimately lead to breaking of the cell membranes and thus the intercellular components will come out of the cell. Phage or viruses that lyse bacterial cells are commonly used for cell disruption. Now in case of intracellular products, after the cell disruption method has been applied, the components are now ready for the stage 1, that is solid liquid separation, whereas the first step in extracellular product recovery is the separation of whole cells or biomass and other insoluble ingredients from the culture broth. Now this solid liquid separation can be achieved through filtration, which is the removal of solid particles from a fluid by passing the fluid through a filtering media on which solids are deposited. These filters can further be classified based on the direction of flow of slurry. First category is the static flow or dead end filtration, that is also surface filtration, where the slurry flows perpendicular to the filtration membrane. These include patch pressure filters such as plate and frame filters and pressure leaf filters. Continuous pressure filters includes rotary drum filters. On the other hand, in cross flow filtration, the slurry flows tangential to the membrane surface. Solid liquid separation can also be achieved via centrifugation, where centrifugal force is the driving force for separation, replacing the gravitational force. For the industrial application, different types of centrifuges are available, such as basket centrifuge, tubular ball centrifuge, solid ball scroll centrifuge, multi-chamber centrifuge, and disc bowl centrifuge. Sedimentation is another settling process where the larger particles settle down at the bottom of the tank based on gravity. In flocculation, the cells form large aggregates to settle down for easy removal. Here flocculants are used that promotes flocculation by causing colloids and other suspended particles in liquids to aggregate, forming a floc. Now, after the solids are removed from the broth or slurry, concentration methods are applied. Common methods for concentration includes liquid-liquid extraction, where the separation of a component from a liquid mixture is carried out by treatment with a solvent in which the desired component is preferentially soluble. Aqueous two-phase extraction involves the formation of two immiscible aqueous liquid phases using phase-forming agents such as polymers or salts. The target compound usually partition between the two liquid phases based on their affinity for each phase. Precipitation causes soluble compounds in a solution to become insoluble by adjusting the pH, temperature, or the concentration of the precipitating agent. Now, if we have used solvents for the extraction purpose, evaporation is then used for solvent recovery processes, and it is frequently employed through a distillation plant. This can be carried out by using batch distillation, where the separation of components is done batch-wise, whereas in continuous distillation, the separation of components in a mixture is done using a continuous process. There are no interruptions for this process until the completion of the distillation. Supercritical fluid extraction is a process of separating one component from another using supercritical fluids as the extracting solvents. A supercritical fluid is any substance at a temperature and pressure above its critical point, where the liquid and its vapor do not exist as distinct phase, rather the liquid and vapor can coexist. The next stage is purification, which is again a critical step in downstream processing for removing impurities and isolating the target product in a highly pure form. This can be achieved using chromatographic techniques which are used to isolate and purify metabolic products. Adsorption chromatography is based on differences in adsorption at the surface of the solid stationary medium. Ion exchange chromatography allows the separation of ions and polar molecules based on the charge properties of the molecules. In gel filtration chromatography, the separation of the particles is based on their size, shape, and molecular weight. Affinity chromatography is based on the property of specific and known covalent binding of proteins to other molecules, which are referred to as substrates or cofactors. In high-performance liquid chromatography, the separation is greatly improved by applying high pressure. Partition or liquid-liquid chromatography is based on the differential partitioning of compounds between a mobile and stationary phase. Membrane processes includes reverse osmosis and ultrafiltration, which utilize semi-permeable membranes to separate molecules of different sizes. Liquid membranes involve the selective transport of solutes across a liquid phase, acting as a barrier between two immiscible phases. Emulsion liquid membranes involves the use of an emulsion. In supported or immobilized liquid membranes, liquid is immobilized in the pores of a thin microporous solid support. In bulk liquid membrane, the liquid membrane phase is typically immobilized in a porous support material. Crystallization method is used to obtain pure crystals of the desired substance from an impure mixture. After purification has been achieved, finishing or drying is the final stage of product purification and formulation. It involves removal of moisture or solvent from a substance, resulting in a solid or dry product. So drying can be achieved by any of the following methods, that includes drum dryers, where the heated surface is the envelope of a rotating horizontal metal cylinder. Spray dryer, that includes atomization, droplet to particle conversion, and then particle collection. Fluidized bed dryers are also used to reduce the moisture content. It works on the principle of fluidization of the feed materials. And finally, the freeze drying or lyophilization, where the material is first frozen and then dried for removal of ice or other frozen solvent in a high vacuum by sublimation. So this was the overall downstream processing that has been categorized into different steps for better understanding. For details of all these methods, you can go through the detailed presentation that were discussed in earlier sessions. Thank you.